Next up is Dr. Debra Bangasser. Dr. Bangasser is an associate professor of psychology and the director of the Neuroscience program at Temple University, and she's also the principal investigator of the Neuroendocrinology and Behavior Laboratory. Dr. Bangasser's research investigates sex differences in stress responses and their contributions to disease vulnerability and resilience. And she's also interested in how early life experience can promote sex-specific resilience to alterations in cognition and motivation. Dr. Bangasser has won numerous awards for her research, including the Janet Rosenberg Trubatch Career Development Award from the Society for Neuroscience and the American Psychological Association Presidential Citation. Tonight, she is joining us to present her talk entitled Male and Female Brains Under Pressure. So, Dr. Bangasser, thank you again for joining us. Thanks for bearing with that exceptionally smooth transition, and I will turn the presentation over to you.
Thanks. I've never been introduced with gestures before. It was great. So, all right. Well, thank you so much for having me. This is such an exciting event to be a part of. I'm really, really thrilled to be here. And my lab at Temple University is a rodent lab, and we use rats to make important discoveries about the brain. And research that uses rodent models to develop treatments to improve human health is called preclinical research. And preclinical research is crucial because there's no synthetic way to grow a brain in a dish. Computers can't model all aspects of brain function. And there are human neuroimaging techniques, which I think you'll hear about in a bit, but they're limited in the molecular changes that you can observe in a brain. So preclinical research is crucial to identify factors that promote vulnerability to psychiatric and other brain disorders. And they're also crucial if we want to develop treatments that are safe and effective for brain diseases before testing them in humans. So now that I've told you about the type of research I do, I thought I would tell you what I study. And as Evan mentioned, I study sex differences in the brain. And there are actually a lot of sex differences in the brain, but my lab studies sex differences in stress responses that may bias males and females towards different pathology. But before I tell you some things about sex differences in the brain, I thought I'd ask you guys a question. So we're going to have a poll on the next slide. And I thought I'd ask you a question about sex and gender. So what percentage of the US population do you think is female? And so there should be a poll that you can fill out.
Okay, Debra, the poll is being filled out right now. We have 17 votes. I might give it another minute or two.
Okay.
A couple seconds.
Yeah.
Maybe not a minute. It looks like overwhelmingly 52%.
52%. That is a really good guess. So hold on just a second. My slide somehow disappeared when I clicked on the poll. There we go. So it's actually 51% of the population identifies as female. There's a small portion of the population that identifies as non-binary, and the rest identify as male. So given how important preclinical research is, what percentage of preclinical research subjects do you think are female? So again, there's another poll. And Ashley, I'll let you read me the answers when they come in.
Sounds good. I wonder if you have any lab members in the poll right now.
I do. I might have stacked the crowd.
I think you might have. We are coming in, so we have 70 votes in. 86% of our votes are for 30%.
Well,
30%
so
of the population.
You might guess, one might expect that in preclinical research, there would be about 50% females reflecting the human population. But my lab was right. I taught them something. Indeed, there's only 30% of preclinical research neuroscience studies use female subjects. And just to be clear, when I'm talking about rodents, I'm talking about biological sex. Gender is a psychological construct. So rats and mice don't have a gender. But because we're only using about 30% of female research subjects, it means that we're learning a lot more about the male brain. And this is a study from my colleague John Meitzens's lab. It came out this year. So this is the most recent state of the field. Now, the data was collected in 2017. It takes a while to analyze. But this is neuroscience studies. And you can see that the green portion of this pie is the biggest, and it's studies that only use males. So there is a portion of studies in gray that do include males and females, but sex differences are not compared. So sex is not really considered. Only 5% of studies compare the sexes. And I think this is really important to do because we know that there are sex biases and a number of brain disorders. So in this table, disorders that are more common are towards the top of the slide. In women are towards the top of the slide. And these include things like PTSD or post-traumatic stress disorder and major depressive disorder. Disorders that are more common in men are towards the bottom of the slide. And these include things like Parkinson's disease and attention deficit hyperactivity disorder or ADHD. And even when disorders occur at fairly similar rates in men and women, they can present differently. So schizophrenia, as an example, is only slightly more common in men than in women. But on average, men with schizophrenia tend to develop symptoms sooner, and they tend to have more cognitive deficits than women. There are also some differences in treatment. So for major depressive disorder, a class of drugs called monoamine oxidase inhibitors tend to work better in men, whereas selective serotonin reuptake inhibitors tend to work better in women. So taking all of this together really indicates that there are biological sex differences that can contribute to these diseases. And my lab studies stress. And we're interested in stress because unchecked stress can make symptoms of all of these disorders worse. In the case of PTSD, it's actually precipitated by a traumatic event. But symptoms for all these diseases can get worse under conditions of unchecked stress. And stress can even initiate the bout of a disease. So, for example, depression often follows a stressful event. So we've been really wondering, are there sex differences in stress responses that may bias males and females towards different pathology? And one of my favorite stress modulators is corticotropin releasing factor. I'm going to abbreviate it CRF. It is the same thing as corticotropin releasing hormone. And this is a sagittal or sideways section of a rodent brain. The rat's nose would be over here. And you may have heard of a stress hormone called cortisol. It's a type of glucocorticoid. And CRF was first described in causing the release of that stress hormone. So activating something called the hypothalamic pituitary adrenal axis, which causes cortisol to be released from the adrenal glands. And so it orchestrates
Debra, if you can hear me, you froze a little bit. There you go.
Stress to regulate brain regions. Did I freeze?
You froze for a second, yes.
Okay. I couldn't tell. I look frozen on my photo and a lovely pose. Okay, so if you can still hear me, I'll keep going. Ashley, can you hear me? Okay, perfect. So I'm interested in how CRF regulate brain regions during stress. And this is really crucial because it's the CRF regulation of different brain circuits that's thought to modulate cognitive and behavioral responses to stress. And one target of CRF regulation is the locus coeruleus, or LC. And this is an important arousal system. And so what happens is that CRF is released during stress into the locus coeruleus, which causes locus coeruleus neurons to increase their firing rate, which causes norepinephrine, abbreviated NE, to be released throughout the brain to increase arousal. And you can imagine that in response to an acute or moderate stress, activation of the system is really important. I mean, we want to be alert during a stressful event. However, if this system is activated persistently under conditions of maybe extreme stress, it can lead to a dysregulated state of hyperarousal. And hyperarousal is that feeling of being on edge. It is, oh, I'm back. So it's that feeling of being on edge. Hyperarousal is a key feature of PTSD, and it also contributes to symptoms in certain patients with depression, such as a lack of concentration, restlessness, and sleep disturbance. And as I mentioned, PTSD and depression are more common in women than in men. So when I was a postdoc in the lab of Rita Valentino, who was at the Children's Hospital of Philadelphia, Rita and I were really interested in whether there were sex differences in the way CRF regulates the locus coeruleus. And we found a number of sex differences. I'm just going to go through one here. And so on the next slide, you are going to see a magnification of two neurons within the locus coeruleus. So you have our CRF containing neuron, which releases CRF across that structure that Michael was talking about called a synapse. And then we have our locus coeruleus neurons. And on the locus coeruleus neuron are white structures depicted here, which are CRF receptors, and they bind CRF. And when CRF binds to these receptors, it activates the locus coeruleus neuron. But what I've shown you here is that there are receptors that are on this plasma membrane where they can interact with CRF. And there are also receptors that are localized inside the locus coeruleus neuron. And these receptors aren't static. They can move back and forth between the membrane and inside the neuron. And this neuron is releasing a lot of CRF, so it's a really stressed neuron. And what is thought to typically happen under those conditions is that these receptors move from the plasma membrane inside the cell in a process called internalization. They go internally. And that reduces the effect of CRF on the locus coeruleus neuron. And this is thought to be compensatory to prevent the locus coeruleus neuron from becoming overactivated during a lot of CRF release. And I should say that this process of internalization was characterized, probably not surprisingly, in males. And so I was really curious if this was also occurring in females. And what we found in females is that there's a lot of CRF being released, but under those conditions, instead of the receptors becoming internalized, they're actually trafficked to the membrane. So there was more on the membrane, meaning that this locus coeruleus neuron can become very activated in females under conditions of a lot of CRF release, which would occur during a very stressful event. And remember, if this neuron is activated, it can lead to high levels of arousal. So what do we think this means for the organism? So I like to think about arousal as being on a continuum. We can be asleep, we can be perfectly alert, which I'm sure you all are during this talk, or under really rare extreme conditions, we can get pushed into this dysregulated state of hyperarousal. And so I think these sex differences in CRF receptors in the locus coeruleus shift females a little bit to the right along this continuum. And maybe most of the time, this makes no difference. There could be some circumstances where it's actually helpful to be really alert. But under conditions of extreme stress, extreme CRF release, females may be more likely to shift into this dysregulated state of hyperarousal. And if similar mechanisms are occurring in humans, it may explain why women are more likely to suffer from psychiatric disorders with hyperarousal features. So my lab at Temple has focused more on how stress and CRF can regulate cognitive processes. And we're interested in cognition because it's dysregulated in a number of psychiatric disorders, including schizophrenia and ADHD, which are more common in men than in women. And one circuit that we've investigated is, again, this is a sagittal section of the rat brain. Rat's nose is over here. We've looked at a circuit that's involved in memory. So if you've heard of any brain region related to memory, you might have heard of the hippocampus. And the hippocampus receives a projection from a brain region called the medial septum. And this circuit regulates certain types of memory. And my former graduate student, Kim Wasalis, was really interested in what CRF was doing in the medial septum. And she suspected that it might have a role there because the medial septum has a high density of CRF receptors, meaning that it's a target of CRF regulation. But no one had studied how CRF could regulate the medial septum before. So what she did is she put CRF in the medial septum and tested rats on a learning task. And the task she used is the object location task. And in the object location task, there's a training phase where rats are put in a chamber with two identical objects. They have time to explore the objects and the chamber. Then they're removed from the chamber and one object is moved or displaced. Now, rats are curious creatures. So if they recognize that an object is moved, they'll spend more time exploring the displaced object. And when they do that, we say that they've formed a spatial memory, an object location memory. And what I'm going to show you on this graph is on the y-axis is a ratio of the time the rat spends with the displaced relative to the familiar object. So a ratio of 1 means that they're spending the same amount of time with both objects during the testing phase, and so they really haven't formed that spatial memory. So on the x-axis, we have different doses of CRF that were put into the medial septum, and you can see that the high dose of CRF impaired this location memory in both males and females. They're in the triangles on this graph. But what was remarkable is that in the medial septum, the low dose of CRF only impaired memory in males. So in the medial septum, this memory region, males were more sensitive than females to CRF. And so now our lab is trying to figure out either what makes males vulnerable to the CRF in the medial septum, or actually what we're pursuing, because we have some data to suggest that this may be the case. There are factors that may make females resilient to this low dose of CRF in the medial septum's effect on memory. So one protein that can regulate CRF function that I haven't discussed yet is a protein called CRF binding protein. And what you need to know about this is the more binding protein, the less CRF is available to interact with these receptors and activate the septal neuron. So what happens when CRF is released, if binding protein's around, some of that CRF goes to the receptor and activates the septal neuron, but some of it also goes to the binding protein. So if there's more binding protein, there's less ability to CRF to activate the septal neuron. And in collaboration with Matt Wimmer and Lexi Ellis, we looked at CRF binding protein expression in the medial septum and found that females had more CRF binding protein. So what we think is going on is that this is helping protect females from the memory impairing effects of a low dose of CRF. So to summarize the data that I presented, when we're talking about the locus coeruleus arousal system, there are sex differences in CRF receptors that make females more sensitive to CRF. And this, under stressful conditions, may make it more likely that females shift to a dysregulated state of hyperarousal and may account for their higher rates of PTSD and depression. In the medial septum, we found that males were more sensitive to the memory impairing effects of CRF. And that means in disorders that have cognitive deficits exacerbated by stress, like schizophrenia or ADHD, this might bias males towards some of those deficits. But what I hope I've shown you is that by comparing the sexes, we can identify factors that promote vulnerability and resilience to stress. We wouldn't think about receptor internalization or binding protein as important factors in these disorders if I only looked at males. And as a reminder, this is the current state of the field. we're mostly only looking at males. What this means is, given that preclinical research is how we develop treatments for disorders, it means that we're developing treatments based on male biology primarily. And so it may not be surprising to anyone that women have more adverse drug reactions than men. We're making drugs for men. And this also means that if we're not looking at females and comparing females to males, there may be therapeutics that would work really well for females that will never make it to market. So this approach of focusing only on males may be denying women some important treatments. And my hope is that in the near future, the survey of the literature will look like this, where males and females will be included in experimental design and compared. And there's some hope that we're moving in this direction. So funding agencies in the US, like the National Institutes of Health, and other countries like Canada are starting to require that preclinical studies consider sex as a biological variable. And this is really crucial because we're trying to move into an age of personalized medicine where we tailor treatments based on individual physiology. And certainly biological sex is going to be one of those factors that we need to consider in thinking about individualized physiological responses if we want drugs to treat brain disorders that work well in all genders. And with that, I have a lot of people to thank. So my postdoc mentor, Rita Valentino, was a wonderful mentor and helped me fall in love with CRF. Lexi and Matt did some of the work on the CRF binding protein. Kim did a ton of work looking at CRF in the medial septum. This is my current lab who are studying all things sex differences and stress. And I'd like to thank my funding agencies. And I'd really like to thank you for your attention and for the organizers for putting this together. I'm really happy to be here and answer questions.
Okay, thank you, Deborah, for the amazing talk. I have a few questions to ask you. The first is from Kendall, who's actually an NGG student here. And she asked, do you think being a mother or a virgin can affect the hyperarousalness of females?
So we have not looked at that yet, but there is a lot of changes that occur following pregnancy. Some are related to the pregnancy itself. Some are permanently changed. Your brain permanently changes after pregnancy. And so we haven't looked at that, but I definitely think that there would be changes in arousal. Certainly we see differences in arousal directed towards pups, but I have a feeling that this would be differentially regulated by CRF too.
Great, thank you. We also have one more question from Joseph Gallegos. What cells are producing the CRF binding protein?
That is a really good question. We don't know the answer to that. We just got a grant funded to look at that. There is so little known about binding proteins, and they're these really cool regulators of molecular function. And so we have some preliminary pilot data that maybe it's GABAergic neurons that are producing it, but that is not ready for prime time. So stay tuned. That's exactly one of the questions we're asking.
Okay, great. Thank you. I think it's time for us to transition on to our final speaker.